Hi, I'm Whitney Espick, the CEO of the MIT Alumni Association, and I hope you enjoy this digital production created for alumni and friends like you.
Good morning. It's an honor to have the opportunity to address you this morning. I am sort of setting the stage for how we view artificial intelligence and information technology transforming biological science and biomedical science. And to do that, I'm going to talk a little bit about history and set the biotech development in the context of this, and then turn to information technology and how I see it transforming the future of medicine and science. If you think about it, this whole subject in the context of modern molecular biology began with the discovery of Watson Crick, Rosalind Franklin, and Morris Wilkins in the structure of DNA in 1953. Now, the interesting thing about this is that shortly thereafter, in the late 50s, Jim Watson was recruited to Harvard to start a group of molecular biologists at Harvard, and Jim Watson's mentor, Salvador Luria, was recruited to MIT to convert the biology department at MIT into a gene and molecular-oriented department. So here we see at the two ends of Mass Ave, these universities making a transformation in biological science, orienting this science to genes. Now, it took us about 10 years to understand how information from DNA was transmitted into the cell from the biological process that's inherited from each individual. And what you see at the top of this slide is a schematic of DNA, this beautiful molecule that's long and linear, and you know the nucleotide sequence of that DNA has the information for biological function. And it's transmitted as information into the cell by an intermediate called RNA, and then that RNA is taken and decoded by a biological machine to make the proteins that carry out most of the functions in cells. So this whole information transfer process was elucidated for the first time in the early 60s, mid-60s, and told us how DNA then was decoded in the cell to carry out function. All of this was work done without mostly chemistry, and was done with a pure intellectual interest of understanding what DNA was about. But knowing how to manipulate DNA was a big part of this science, and also set the stage for what happened in the middle of the 70s, where there was a true revolution in chemistry and biological science with the development of recombinant DNA, the ability to take DNA and join it end to end in specific ways, the ability to sequence DNA, and this was Wally Gilbert at Harvard and Fred Sanger in Cambridge, and the ability to chemically synthesize DNA. And this really MIT had a major role in, because Gobind Khurana, the person who first synthesized a gene, was a faculty member here at MIT in the Department of Chemistry. So then in the mid-70s, we had the ability to do synthetic biology, to actually make gene structures of novel function, and then to carry out the introduction into a cell to see the activities of new cells. And this was truly transformative. And then there was this process of biotechnology, because we recognized that it was possible to use this chemistry to solve major societal problems, and in transferring the science from the laboratory into private companies so that it could grow was the origin of biotechnology. And here MIT had some very special relationships. Bob Swanson was the founder of Genentech with Herb Boyer. Bob was a chemistry undergrad at MIT and a Sloan School graduate. He worked with Kleiner and Perkins, a venture capitalist on the West Coast. Tom Perkins was an MIT graduate. And the third company established was Biogen here in Cambridge and Geneva, Switzerland. It was established in 1978, and the impetus for getting involved in establishing this company actually was an MIT alumni, Ray Schaefer, who's sitting in the middle of this picture. And this is a picture of a Biogen organization early in '78. That was a founding year. It's 40 years old now. And myself on the left side, Wally Gilbert in the middle, and a number of European scientists. It was originally established in Geneva, Switzerland, and the motivation here was to bring the science that we had developed in the laboratory into society so that it could grow. It was mind and hand at its most fundamental level. And this has advanced enormously over the years and has had a transformational impact, not only in medicine and in science, but also in the economy and employment around the world. What I'm showing you here is Boston and MIT and Cambridge. And if you see this, or in looking at this, you're looking down from Harvard, from the north to MIT. MIT is these red buildings along the Charles River at the bottom of the slide. And what you note around MIT is a number of blue buildings. These are all biotechnology pharmaceutical companies. They range from Biogen, who has 6 buildings in this space, to Novartis, which has its international headquarters for pharmaceutical science here in Cambridge, about 1.5 blocks from us. There are about 70,000 jobs in Massachusetts now that are related to biotechnology. 35,000 of those jobs is research and development. It is the most intense research and development community in life science in the world, and it is organized along the red line on the boundaries of MIT, with Harvard at the top and Mass General and the hospitals here at the bottom. So over something like 50 years, this science has transformed our environment here at MIT and in the country and the world. Now, if you want to see that in graphic terms, I will show you. This is Kendall Square in 1975. I came to MIT in 1974. And this is what the Kendall Square area. Kendall Square is down to the upper right there. This is Draper Labs. You're looking to the south. And it was vacated because NASA was hopefully going to move here, at least the research station. And this part adjacent to MIT was emptied by Cambridge for that reason. Biogen started in a small building over on Benty Street in the upper left. If you look at Kendall Square now, and this is 2012, and I know this is like your TV commercial for your cosmetics. It's black and white, and then it's colored. But it does project the issue that here's Kendall Square with Draper here at the bottom, again, in the same orientation, Kendall Square up to the right, and all of this around us and swooping around the campus in the other direction is the biotechnology pharmaceutical community here, and it is also a community that has energy research in it, IT research in it. We're going to talk a little bit more about that, but it is the hub of biomedical research innovation. I did a calculation, and in my mind, 35,000 people in the biotech community doing life science in this community, and there's at least 20,000 on the campuses of students and graduate students and postdocs and faculty, which sums to 50,000 people in this community doing life science research now. So how did this come about? It came about because of the investment and intellectual nurturing of the life science on this campus. And I just want to point that out to you because many of you have been part of that over many, many decades. In 1972, the Center for Cancer Research was established here, doubling the size of biological science and cell biology. Whitehead was established 10 years later in introducing 20 new scientists in life science. Biology as a core requirement, spreading the language of biology and molecular biology through the campus was established in '93. In 2000, we expanded neuroscience by a new department and the establishment of the McGovern Institute and the Pickhour Institute here on campus, turning the campus into one of the outstanding, if not number one, neuroscience communities in the world. In 2003, the Broad Institute, which I'll say a little bit more about in a few moments, was established, ultimately came into MIT as part of MIT for several years, and then 10 years ago became freestanding from MIT with a relationship to MIT and Harvard when Eli Broad committed about $500 million to endow this institute here on the edge of MIT. And the Koch Institute was established in 2010. And the Koch Institute is a cancer center institute where engineers and biologists on every floor are working together to attack this problem of cancer, and it has been extraordinarily successful. And the IMEDS Institute was established after this. I'm sorry I didn't have it on here, to be a bigger bridge between engineering and life science and the hospitals at MGH and Brigham and Women's. The engineering department at MIT has been a deep partner in this over the last decades with the establishment of the Department of Biological Engineering here at MIT, one of the most thriving departments on campus. Now, a third of all MIT engineers are involved in life science, and that sets the stage for this new informational technology transformation that President Reif was talking about earlier today. And as I just mentioned, MIT and Kendall Square and biotech and information technology around us is not a one-way street. The MIT is influencing that community, but that community is influencing MIT. And we have a deep partnership with our communities here, advancing life science in ways that is transformational, fundamentally transformational across the world. So I want to talk a little bit about the Broad Institute because it sets the stage in many ways for information technology being a deep part of the future of biomedical science and medicine itself. Broad Institute grew out of the national initiative to sequence the human genome, and Eric Lander in the 90s established the Human Genome Institute. Eric Lander, a professor here at MIT, and an institute investigator at the Whitehead Institute, established the Human Genome Institute here at MIT. It has grown over those years. The sequence of the first human genome was announced as complete in 2003, the 50th anniversary of the discovery of the structure of DNA by Watson and Crick. It cost us $3 billion in 15 years to get that sequence. It was mostly about how to develop the technology to do that in a rapid way. And if you think about it, it is an astounding problem. The amount of DNA in your genome, if laid out end to end in every cell, is this long. 3 angstroms as a step, 1 billion units, it's this long. And you had to determine the sequence of all of it exactly. We have that technology now. It doesn't cost us $3 billion, and it doesn't take 15 years. It costs us less than 1,000, and it takes hours. We have really, by technology, transformed how we obtain information and use it to understand life science. So the Broad Institute here across the street and the Human Genome Initiative from which it grew was the major contributor to this science of genomics. There's only 2 major institutes now that I see in the world that are competing academically and that is the Broad here and the Wellcome Trust in England, and we are a leading force in this. Does it matter? So I'll give you a trivial example. This is by sequencing human cancers. This is an indication, as listed on the left, of how many mutations are in individual cancer cells. So on the left, here to the left is colon with a DNA repair. And in those tumor cells, there's 1,000 mutations in those cells. 1,000 genes have mutations in them. That's really striking because you only express 10,000 genes. So 10% of them have mutations in them. That cell is so foreign that if you modulate the immune system by immunotherapy, you get frequent elimination of the tumor cells. Even if they're disseminated through the body, it is a remarkable new therapy. The next three are lung cancers and melanoma. They have about 150 mutations in genes. 150 mutations. Where do they come from? Smoking and sunshine. And those tumors frequently, when treated with immunotherapy, are seen as so foreign in your body that they can be rejected by your immune system. The other tumors listed through adult solids have about 50. Now, they don't respond very well to immunotherapy, and we're trying to figure out why and how to manipulate it. And information technology is going to be an important part of this. And over to the far right is pediatric tumors, and they have very few mutations in them. So we don't expect that immunotherapy is going to be a major advance or use in those tumors. So I've described where we're at in terms of genome and molecular biology and cell biology in a very broad way. What's the future? And the future is something we've called here at MIT convergence. It's advances in information technology, material science, imaging, nano, and optics, coupled with computing, have transformed the physical world and created all these devices and the way we communicate and the way we live. They're now ready to begin to transform life science. The stage is set. We need this technology, and in fact, it's a major part of the future of all medicine. What we need to be able to do is to integrate a large amount of information to understand the process of disease and how it develops over age and how we can intervene. We need medical histories. We need to know something about the environment. We need to know something about behavior. We need medical data from imaging and metabolism and cellular status. And we need genetic profile because we don't all have the same risk to developing different cancers. We, by our nature of inheritance of different types in different places in the world, have a different susceptibility to cancer. And I will comment on that in a moment. So this is what we want to be able to do, and MIT has just recruited a young scientist in engineering, EECS, who will quickly become a leader on campus, Dave Sontag, is working on this, and that is taking medical records and other information and being able to identify before you have a diagnosis in a conventional sense, before the disease needs to be treated, the susceptibility, the risk to developing a disease. Because if we can identify those individuals, we can frequently change lifestyles, frequently change medicines slightly, and reduce the risk of advancing into a condition of medical intervention and treatment. Regina Barzali, which you heard mentioned earlier by Anana, has been working on this problem from the perspective of breast cancer. And I want to use her work in the next few moments to illustrate how this could be so profoundly important. What we do now for current diagnosis of breast cancer is we do pathology and we do imaging. We reduce that to a text. Text can be very elaborate or very brief. And then we say it's a grade 3 estrogen, progesterone negative, HER2 negative tumor. We can do grade 1, grade 2. There's about 6 or so different diagnostic outcomes, and then we do a treatment. So we have all this information, but we reduce it to a very simple protocol. And why? Because the way we do our science, we identify particular diseases, as I'll mention in a moment, and treat them in particular ways. So we know that these grade 3 different tumors have very different outcomes, even though they have a pathology and histology that is reduced to those same information. If we could bring more information to this, could we distinguish those individuals who have a more rapidly advancing and critical disease from those individuals who may never see that disease again, or it's quiescent through their life? And this is a question of how much information you can bring to distinguish subclasses of these diseases. So what we conventionally do now is carry out clinical studies and classify these tumors from papers that are defined studies and responses to diseases and progression. But if you take medical records, in this case it's medical records out of a collaboration by Regina with Mass General of 6,000 patients, it could be 60,000 patients, it could be 6 million patients. That type of information is going to be available. And with that information, could you subtype these tumors for better diagnostics and therapeutic treatment? Now, one of the categories you're going to want to add to that is the genetic constituents of the individuals who are being diagnosed. And this is a study that was, in fact, just being published out of the Broad Institute. It's using English data, and I'll come back to that in a moment, but it's using data that you have a polygenic score. You look at the mix of genes in an individual, and you ask, what's the risk of that individual due to their mix of genes developing breast cancer? And what you see here from 0 to 100%, and that's the category of prediction of risk using genomic complexity and the prevalence of breast cancer, that if you move over to that 90% point, those individuals have a 4-fold increased risk of developing a critical breast cancer through their life. If you move to the 99%, 1% out of 100, 1 out of 100, they have almost a 5 to 6-fold increased risk. So by looking at the genetic makeup in a population, the sort of data 23andMe can now capture, you can get a risk factor for different types of cancer. So if you combine that information for thousands of people with better machine learning analysis of images like MRI images and CAT scan images, better analysis by machine learning of histology and pathology, capturing tumor cells, which is shown up at the right, and looking at the genes they're expressing. They're expressing 10,000 genes, and it varies enormously from one tumor cell to another. So they give you an enormous amount of information. And then using genomic profile, using that information, it is almost impossible that we will not be able to identify individuals of higher risk or lower risk, intervene earlier with more specific agents to get better outcomes. And this is true in breast cancer, but it's also going to be true in every other type of cancer. We've decreased the rate of death due to breast cancer by 1/3 over the last 20 years. We can drive that down to very small numbers with this type of information, and that's true of many other cancers. So what we need is an enhancement of science of cancer with better intervention and prevention by this type of information, providing biomarkers for cancer that we can monitor, follow, monitor, and treatment with. And we can enhance communication between the physicians and the caregivers and the patients because we'll know more and we can communicate with more information to help them. Now, all of this technology seems available. Why aren't we doing it? There are actually two major reasons. Research in this country is funded on the treatment side. The National Institute of Health, which is a wonderful organization, has transformed biomedical science, but basically focuses on disease processes and how to intervene in disease processes. Pharmaceuticals are developing new treatments. There's not a lot of investment on how to predict risk and intervene earlier in the risk process of disease progression. And that's because the payer side, the insurance side, has not taken this on as a task. And then the second and last thing I will mention is that we have a very integrated healthcare system in which the payers in one place, the healthcare providers in another, patients in another. We're not an integrated system. And then dealing with patient privacy information becomes much more difficult here. So we're going to have to overcome those to see this turn into a real science. MIT will be a leader in it, and it's going to be very exciting. Thank you very much.
Thanks for joining us. And for more information on how to connect with the MIT Alumni Association, please visit our website.